electricity. And instead of calling it electricity, when we're talking about neurons, what do we call it? What's the flow of electricity through a neuron called? You may not have been there yet. You're going to hear called an action potential. It's just an impulse. It's the flow of electricity through a nerve. Now, another important thing to think about, if this stayed there, if this receptor stayed bound to this channel, what does that mean about the channel? It's going to always be open. What has to happen to those neurotransmitters right after they cause their response to happen? They have to be gotten rid of. So they're either broken down in the synapse or they're pulled back into that presynaptic neuron. That way, once it's removed, what happens to the channel? It closes back up and it can refire again seconds later. All right, so let's look at question 3. Go ahead and take a few seconds to try to answer question 3. This one should be easy. So what do you got on question 3? A. Because testosterone is a steroid, and a steroid is a lipid-based hormone, right? It can go through. It can just as easily be estrogen. Either one work. They're both steroids. And remember that the steroids penetrate the cell membrane. They bind to a receptor on the inside of that membrane. We see that right here. They then go through the nuclear envelope. They bind to a specific gene, activate that gene, cause it to be transcribed and translated into a protein. And they tend to cause slower but more sustained responses than ligands that bind to extracellular receptors. So there's question 4. Go ahead and answer that. You may struggle with this one. What do you guys say on this one? I heard D. I heard B. All right, the correct answer here is B. Let's look at each choice here for a second, though. Hormones are chemical messengers that travel through the circulatory system, right? That's what makes a hormone a hormone. There's a whole session today on feedback loops. Most hormone concentrations are regulated by feedback loops. Hormones often regulate homeostasis through antagonistic functions. Take blood sugar for a minute. What does insulin do to your blood sugar? It causes it to lower it. What does glucagon do? Raises it. What does antagonistic mean? They go against each other. There's a lot of hormones that occur in what we call antagonistic pairs. One does one thing, the other does the opposite thing. But together they always keep you where? Balance, near the set point, near the homeostatic set point. So B, hormones of the same chemical class usually have the same function. Steroids are a chemical class, right? Testosterone and estrogen belong to the same chemical class. Do they cause the same response? No. If you were a bodybuilder and you were going to cheat by taking steroids and somebody accidentally starts giving you estrogen instead of testosterone, is that going to work out for you? Probably not, okay? Probably not. So B is what we're looking for. All right, let's go ahead and look at question 5 as well. Go ahead and answer question 5. All right, something's not numbered right in the packet. My question 5 is your question 6. So you guys go ahead and do question 6. Yeah, we'll come back to 5 later. Just go ahead and do 6. So what do you guys say in this one? C. This is another case, and this is kind of a theme in AP biology, theme of common ancestry. If you share the same proteins with other different kinds of living things, what else do you share with them? If we share the same kinds of receptors between us and, let's say, a snake, how did we get those same proteins? We had to have what? What codes for your proteins? Yeah, I heard it. Nice. DNA, which means somewhere down the line you shared what? Common ancestor, somewhere down the line. Many of the cell signaling pathways are the same throughout all eukaryotes. That's important.
We had a question like that on our test about evolution. Yeah.
Good. So here's one about insulin and glucagon. We kind of talked about this already. This is an example of what? What are we looking at here? When you see that figure 8 kind of thing, what are you looking at? That's a feedback loop. What kind of feedback loop? Try again. It's a negative feedback loop. Okay. And you'll have a whole session on positive and negative feedback loops. By far, what's the most common kind of feedback loop in your body? Negative. Negative feedback loops are always trying to push your body where? Toward what? Toward the set point, toward where it needs to be. Positive feedback loops don't do that at all. And we'll get into that in the next session. So here's the set point for blood glucose. Should be between 90 milligrams per 100 milliliters of blood. So let's say that you ate a healthy breakfast like I did this morning, which was a Mountain Dew and a Little Debbie cake, okay? So packed with glucose, right? Packed with sugar. So I got a sugar rush. My blood glucose shot up. Eventually my pancreas was made aware of that, and it released insulin into the blood. That insulin traveled through the bloodstream to lots of different places, but in particular, the liver, just general body cells. The insulin binds to receptors on those, and what does it tell those cells to do? What does the insulin tell cells to do? Take glucose out of the blood and store it. Maybe store it as fat, if we're talking about a fat cell. In the liver, store it as what? What does sugar get stored as in the liver and in the muscles? What polysaccharide? Glycogen. So because it's being taken out of the blood, what happens to the blood glucose levels? They drop, they get back here. Once they get back here, what does that tell the pancreas? Stop making insulin because we're where we need to be. We're at the set point. On the other hand, let's say you didn't eat breakfast this morning, and you hadn't had anything, and maybe we get to session 3 and you still hadn't had anything. Your blood glucose starts to drop. Your pancreas is made aware of that. It releases glucagon, which travels through the blood, goes to places like the liver, telling the liver to do what? Break down glycogen and release glucose into the blood, raise them back up. Eventually those levels get back where they're supposed to be. What does that tell the pancreas? Stop producing glucagon because I'm where I should be. That's a negative feedback loop. Always pushing you toward the set point. All right, so it looks like I just reversed 5 and 6 in this presentation. So go to question 5 in your packet. Answer question 5. so what do you guys think? A looks good, right? Remember, epinephrine is a hormone. It's a protein-based hormone, so it binds to an extracellular receptor on the surface. It activates that adenylyl cyclase enzyme, which then will start to make CAMP, C-A-M-P, which will then activate what? What does CAMP turn on? Why do we need CAMP? What is this job? To turn on the phosphorylation cascade, right? The signal transduction pathway. So A looks good. So here's question 7. Go ahead and answer that one as well. You'll see a lot of the questions on your AP exam will look like this. There'll be some kind of diagram or picture, and you have to decode the picture. What do you guys think on this one? A looks good, right? Remember, steroids like testosterone are lipids. They go straight through the membrane, which you see happening here. They bind to an intracellular receptor. Together, that receptor and the hormone go into the nucleus, bind to DNA, turn on specific genes. They act as transcription factors for specific genes. So A looks good. All right, so now let's do a math question. Now this math question has absolutely nothing to do with what we just talked about. That's okay. It's just math that we needed to review. So take a stab at the math question. Take about 2 minutes. See what you can do with the math question.
Back to the formula page.
Yeah, there's plenty of space right down below it if you want to do it. And you remember, you got your formulas in the front of your packet. I'm mad about leaving it open. You guys have already been through that.
Oh, yes. We've done
Good.
everything you talked about today. we've done.
Awesome. Perfect. You guys want to know what's going on over here? I want to go ahead and go over this because we've got about 15 minutes and we've got some other things we need to get done. Since they're recording this session, I need to make sure I get through all the parts of this packet. So I'm going to move on and do this. We're looking at water potential. Whenever we're talking about the flow of water, we need to think about water potential. So we've got an equation up there on the board. What do we call that little symbol? Well, it stands for water potential. What's the name of it? Psi. Why did they choose that for water potential? What does it look like? Looks like a trident. Think about Poseidon, water. It works. But our equation is the total water potential is equal to, what's the psi s? Solute potential plus psi p. What's the p? Pressure potential. So the total water potential is made up of both solute potential and pressure potential. So let's look at this problem. So we've got a salt solution, right? Sodium chloride in an open container. The concentration is 0.2. The pressure, 0. What's the total water potential at a temperature of 32 degrees C? All right, one thing that you got to think of first is what kind of solute do you have? In this case, our solute is a what? Salt. How do we know it's a salt other than the fact that it's NaCl and it's table salt? What makes salt be a salt? Yeah, what are salts made out of usually? Yeah, usually though, well, not necessarily just sodium and chloride. Usually it's a what and a what? To be a salt, it's made out of what in the front and what in the back? Like potassium fluoride, that's salt. Calcium iodide, that's salt. What makes all three of those things a salt? There's a metal in front and a non-metal in the back, right? That's what makes a salt a salt. This one's a salt. That's important to know. What's the other kind of solute you might have to deal with in these kinds of problems? Sugars. And it should be obvious if it's a sugar, right? Its name's going to end in what? Ose. Okay? This is a salt. That's key. So we've got another formula we need to think about. And that other formula is for the solute portion of this problem. And it's negative what? I-C-R-T. Where the I stands for what? What's the I stand for? Ionization constant. If our solute had been glucose instead of NaCl, what would the I be? Well, it'd be 1, okay? Because sugars don't what? When you dissolve a sugar, it doesn't do what? It doesn't break into ions. If one sugar molecule goes in, what's still there after it dissolves? One sugar molecule. On the other hand, if we put a formula unit of salt into the water, what happens when we put that NaCl into the water? It breaks up into a what?
2 ions.
Yeah, an Na plus and a Cl minus, right? Because it breaks up into 2 pieces, what's the ionization constant for NaCl? 2. Okay? So negative 2. What's the C stand for? The concentration, right? The molarity. Well, that's given to us. 0.2, right? 0.2 big M. What does that big M actually mean? Molarity. What does it mean? What does molarity actually measure? How many moles are dissolved per what? Moles per liter. And I'm going to write, instead of writing a big M, I'm going to write it as moles per liter. And you'll see why in a second. What's the R? It's the pressure constant. And what's the value for that? And it's kind of got some weird units on it, right? What are the units? Liters times, this is on your formula sheet, by the way. Liters times bars over moles times Kelvin. So strange units, that's on your formula sheet. And then the T, temperature, for this to work, it's got to be what kind of temperature? Ooh, I heard 2 things. I heard Celsius and I heard Kelvin. It's got to be Kelvin because of that Kelvin on the bottom of that constant, right? So what is our temperature here? It's 32 Celsius, so we need to put in 305, right? To go from Celsius to Kelvin, you add 273. So we're going to put in 305 K. We're going to cancel out the units that we can cancel out. The K's cancel, the moles cancel, the liters cancel, and we're left with bars. We multiply across. Somebody tell me what the math comes out to be here.
Negative
10.14. So we're just going to say negative. And don't forget about that negative. Negative 10.1 bars. That's the solute potential. So that's going to go right here. What was the pressure potential? Read the problem a little closer. Tell us the pressure potential is 0. So this is 0, which means that the first answer to our question, the total water potential, is what? Negative 10.1. That's the first answer. Everybody okay with that? I think the second question is more the kind of thing you might see on an AP exam. If a plant cell with a total water potential of negative 4 bars is placed into a beaker of the solution described above, will water move into or out of the cell? Justify your answer. So we've got a cell, like so. And here's the water potential was negative 4. We put it into this solution that we just calculated. The water potential out here is negative 10.1. So, how does water move in terms of water potential? But in general, how does water move in terms of water potential? Water moves from higher water potential to lower water potential. So where's the water going to go? I'm hearing inside, I'm hearing outside, because some of you don't know how to deal with negative numbers. Which is bigger, negative 4 or negative 10? Negative 4 is a bigger number than negative 10, right?
Yes.
Think of it like it's on a number line. Negative 4 is closer to being positive than negative 10 is. So that means water's going to move out. And if this were a plant cell, what would happen to it?
You would,
What's the word? Starts with a P.
shrivel up.
Yeah, there's another word though, a big long word. Plasmolysis, which means it would shrivel up. Okay?
And if you wrote that, you would get some.
Then you'd be okay. Right, you don't have to know plasmolysis, but it would shrivel up. Does this make sense?
Yes.
All right, let's go ahead and look at the writing part of this real quick. So we're going to have to move pretty fast.
They've done this exact question.
Good. That's good. This one's off of 3 or 4 years ago off the AP exam. You want this kind of free response on the AP exam. This is one of the short free response questions. Typically, to answer one of these, a good paragraph's probably enough. Make sure that you address the verbs. This says explain the role of each numbered step. So make sure you explain. Don't just say step 1 is, what is step 1 by the way? What's happening in step 1? Reception. What's happening in step 2? Transduction. What's happening in step 3? Response. It's those 3 stages. But it says explain. So you should say for step 1, the ligand binds to the receptor. The receptor does what? Changes shape and moves the message from outside to inside. Step 2, you could talk about there's this chain of proteins that phosphorylate each other. The signal gets what during step 2? It's amplified and spread throughout the cell. At the end, triggering step 3, which in this case, what's happening in step 3? Looks like a gene is being turned on, right? A gene is being transcribed and probably translated to make what? What do genes make after transcription and translation? Proteins. Cause traits. All right, so that's enough on that one. Let's go ahead and look at the long FRQ because we've got about 4 minutes. So instead of having you write this out, which is what I would normally do, let's just go through it together. So this one was off of the AP exam maybe, I don't know, 8 or 10 years ago. Homeostatic maintenance of optimal blood glucose levels has been intensely studied in vertebrate organisms. Part A. Pancreatic hormones regulate blood glucose levels. Notice the verb here is identify, which means what? When they say identify, what do you need to do? Just say it, okay? So identify 2 pancreatic hormones. So if you just said what? Insulin and glucagon are 2 pancreatic hormones that regulate blood glucose levels. You probably got 2 points just for knowing insulin and glucagon and saying. Now you do have to say it in a complete sentence, but just say it. And then B, describe the effect of each hormone on blood glucose levels. So you could say insulin works to do what? Lower blood glucose levels, whereas glucagon works to raise blood glucose levels. If you said all of that, you probably got 4 points on part A. That's easy, okay? There's always going to be on these long free response questions a couple of points that are easy to get. Those are the easy ones to get. Part B. For one of the hormones you identified, identify one target cell and discuss the mechanism by which the hormone can alter activity in the target cell. Talk about reception, transduction, and response. So let's do insulin. So we might say insulin's released by what organ? The pancreas. It travels through the blood. Let's say we use a liver cell as an example of a target. So it binds to a receptor, one of those RTKs on the surface of a liver cell. That's reception. The receptor changes shape. It moves the message inside. It triggers the signal transduction pathway. That's transduction, which amplifies the signal. Ultimately turning on enzymes inside that liver cell that will do what? Break down glycogen and release glucose, thereby doing what? Raising the blood glucose level. That's what they wanted you to talk about in B. And then in C, compare the cell signaling mechanisms of steroid hormones and protein hormones. The keys here. Protein hormones have to bind where? Receptors where? On the outside. Extracellular receptors, because proteins are what kinds of things? Big and polar, charged. And because they're binding to those extracellular receptors, they can cause pretty fast responses, but that are what kind of response in terms of duration? Pretty short-term things. Whereas steroids go straight through the membrane, bind to an intracellular receptor, go to the nucleus, turn on what? What do steroid hormones turn on? Genes. Transcription of specific genes, causing what kind of response? Slower, but more what? Sustained, longer, more long-term. So this is what the rubric looked like for this FRQ. So notice if you identify the hormone, you got a point each. One for glucagon, one for insulin. There's also another one called somatostatin. Probably didn't talk about that one. You also got a point each for simply saying decreases or increases blood glucose level. So 4 points on part A, just as we mentioned. Part B. So you had to identify a target cell. You got to talk about reception. Notice it kind of everything that we talked about, it walks you through there. Transduction, talking about the cascade, the amplification. You can talk about the secondary messengers, and then some kind of response that raises glucose. They had to be really vague with the rubric here because you could have literally talked about hundreds of different kinds of cells in your body because insulin affects lots of different kinds of cells. And then in part C, as we said before, talking about the steroids bind to intracellular receptors, cause slow but sustained responses, typically turning on the transcription of a gene, whereas the protein-based ones bind to extracellular receptors, causing faster but less long-term kinds of responses. And that is session 1. Does that make sense? All right, I hope that helped.
Go take a break. They've got food for you out in the hallway. Need help?
Bump you with Is it in your purse? Is it in your purse?
It's fine, leave it.
Leave it? You good? Who's